Hi, my name is Chaitra Rao. I'm a postdoctoral fellow in MD Cells Lab, and I really want to thank the Sugar Science for showcasing our work that we do at IU. And I'm really excited to talk about our project, the role of EVPDL1 in T1D dynamics. Uh, Extracellular vesicles, for those of you who don't know, are these tiny nanoparticles that are secreted by almost all the cells, and they contain molecular cargo such as DNA, RNA, and proteins. And the EVs there are three different types of EVs. You have the small EVs, which are usually are formed by the endosomal pathway, which range from anywhere about 50 to 200 nanometers. And then you have the larger EVs, such as the microvesicles and the apoptotic bodies that are formed by budding of the plasma membrane. And what's important about these EVs is that their content actually reflects the state of the cells, state of your parent cells, which makes them really beneficial to study because these EVs are stable in extracellular fluid, which makes them a potential biomarker to study the state of a disease. And also, they can communicate in their local environment and also systemically by interacting with other cell types such as immune cells, and potentially they can modulate immune response by negatively reducing immune cell activity or by activating it. And so this brings to the molecular cargo that we are interested in studying, which is PDL1, or which is short for programmed death ligand 1. So program PDL1 is a ligand that is usually present in most of the cells, and it interacts with its receptor PD1, present on immune cells to negatively impact T cell activity. And it's mostly studied in the context of tumor cells, wherein the tumor cells express PDL1 to evade immune response and immune surveillances. And so blocking antibodies against this PD-L1 have been used in cancer therapy, has revolutionized cancer therapy. However, certain individuals that are administered this drug have shown to develop certain side effects such as autoimmune diabetes. And so this kind of shows a specific and a beneficial role of PD-L1 in maintaining pancreatic islet homeostasis. And so this also has been studied by several groups, and shown here is one study from our collaborator in this is Rick's group, where they showed that in early time points of type 1 diabetes, you see an increased elevation of interferon alpha signature, and they found that interferon alpha upregulates PD-L1 in the islets of these individuals. And they also showed that in long-standing diabetes, surviving beta cells that contain high levels of insulin show, or which are positive for insulin, show upregulation of PD-L1. And so this kind of supports the idea that beta cell PD-L1 may be important to protect the beta cells from immune cell destruction. And so we were interested to understand the role of EVs, beta cell EVs in this context. And so our hypothesis of a project was that beta cell EVs may carry PDL1 in their cargo, and that this EV PDL1 may be important to interact with immune cells to decrease their T cell activity. And so to do this, we look to isolate EVs from cell culture supernatant and also from human plasma samples. And so depending on the sample, we either use sequential ultracentrifugation, where you pellet the EVs, or we use size exclusion chromatography if for low volume of the sample to actually enrich EV preparations. And so first, we were interested to see its association of PDL1 with tetraspanins, which is a commonly associated protein in the EV, such as CD63, CD81, and CD9. And what we saw that PDL1 actually co-localized with CD63 and CD81 shown here in human beta cell lines, such as the induced C cells. And then we also went to isolate the EVs from these cells. And what we show here is that upon treatment with interferon alpha, we see that in intercellular, we see an increase in the amount of PD-L1 in the cells. And coincidentally, we see an increase in the EV-PD-L1 levels upon interferon alpha induction. And shown here are CD63 and CD9 that shows the enrichment of the EV fractions from these preparations, and also the lack of calreticulin, which is the EV marker, shows how pure the EVs are, EV preparations are. And we also went to look into the EVs that are secreted from human islets. And we see that consistently there's an increase in EV PDL1 levels upon interferon alpha induction. And we also looked at if this is a non-specific effect of the treatment. And so when we looked at the particle numbers, we saw that there was no, using a nanoparticle tracking analysis, we see that there was no significant increase in the amount of EVs released from the cell upon interferon alpha induction, which shows that there is a specific enrichment of PDL1 cargo in these EVs. And so the next important question that we asked is whether the EVs, whether the PDL1 on the EVs is actually present on the surface of the EVs, because that would allow the PDL1 to interact with its receptor and affect T cell function. And so to do this, we looked at a technology called, we look to use a technology called ExaView, where these contain small microarray chips that are coated with the tetraspanin proteins such as CD63, 81, and 9. And these are used to capture the EVs and immobilize them. And then we can use secondary antibodies such as PDL1 to detect on the surface of the EVs. And so here is the data showing from human islets. And what we see that with either treatment with interferon alpha or interferon gamma, we see an upregulation of PDL1 on the surface of the EVs. And then we also went to show that this EV PDL1 can actually bind to PD1 using an in vitro competitive binding assay with tagged PDL1 and PD1 proteins. And as shown here from EVs from endo C cells, we see dose-dependent change in the EV PDL1 binding to PD1, showing that it has the capacity to bind to PD1. And so the next obvious question would be, what is its impact, or what is the impact of EVPDL1 on T cells? And so to do this, we collaborated with John Piganelli's lab here at IU. And what we did with them, we isolated splenocytes from NOD mice. We treated them or stimulated them with anti-CD3 and CD28 antibodies. And then we mixed these splenocytes with EVs from NIT1 cells with or without PDL1. And then we assayed the T cells using flow cytometry and looked for proliferation, its activation, and effector responses. And so here is a grading strategy that we used. And so we took in live CD45 cells, which marks the splenocytes. And from there, we looked at the CD8 population, and then we assayed the proliferation using cell trace violet dye. And then we looked at activation markers such as CD69 and CD25. So just to give an idea of the amount of EV PDL1 that is present, what we found that the NIT1 cells do not really express a lot of endogenous PDL1. And so we went on to overexpress PDL1 in these cells, and this is the level of PDL1 that we found on the EVs that we used for the assay. And so looking at the results, first we looked at the proliferation. And here we see that in the control cells compared to the unstimulated, we see a nice increase in the proliferation of the CD8 T cells. And this was similar when we mixed these cells with EVs from NIT1 cells. But then we went on to within the EVs that contained high PD-L1, we see that there was a decrease in the proliferation of the CD8 T cells. And so then we went on to confirm that this is a specific effect of PD-L1 by pre-treating the PD-L1 overexpressed EVs with and blocking antibody PD-L1 blocking antibody, and we see that when we pre-treated these EVs, we see a nice reverse of the suppressive effect of PD-L1 EVs on T cell proliferation. And this is also shown in the line graph over into the right. And we also showed that with using an isotype control, it did not affect the T cell proliferation. And then we also looked at the activation markers, here shown as CD69, which is a known early activation of T cells. And we also looked at CD25, which is a late activation marker. And so here in when we had in the control cells with the stimulation and even with the EVs from wild type NIT1 cells, we see it did not really affect the activation of these cells. But then when we went on to culture them with EVs containing PD-L1, it significantly decreased the activation of T cells. And then we also confirmed that this was a specific effect of PD-L1 EVs by pre-treating them with blocking antibodies, and we see that it was able to reverse the effect. And so we were also curious about CD4 T cell population in the mix. So we also looked at the CD4 T cells, and so here showing line graphs looking at CD4 proliferation, CD69 and CD25 as the activation markers. And we were really encouraged and surprised to see that PD-L1 EVs actually also affect the CD4 T cell population here, as shown in the blue lines that showing the control or the wild type EVs, where you see a nice proliferation and activation. And when we add PD-L1 EVs into this mix, we see, as shown in the red, that there was a significant decrease in the proliferation, suggesting that PD-L1 affects both CD4 and CD8 T cells. And then also from the supernatins of these co-culture, we also looked at effector function responses. We looked at granzyme and interferon gamma, and we see that even the effector T cell effector response was affected when we add PD-L1 EVs into the co-culture, suggesting that the PD-L1 EVs affect T cell proliferation and effector response functions. So to summarize the data so far, we show that beta cell EVs carry PD-L1 in the cargo, and it's induced by interferon alpha and gamma exposure. And then we show that the EV PD-L1 has the ability to bind to PD-1 and suppress CD8 and CD4 T cell function. And so these data are, I think, are super cool, and it's important to show. But what the real question or the relevant question rise is, what is the relevance in human diabetes? And so here we looked at a couple of cohorts and looked at EVs from plasma samples of individuals with new onset type 1 diabetes and compared them with non-diabetic controls. And so, to our disappointment, when we looked at the PD-L1 levels in these EVs, we did not really see a difference in the PD-L1 levels. But this could also reflect the different heterogeneity or the heterogeneity in these samples. So we wanted to stratify and look at any type of markers that we can detect to see if there's a correlation between them. And so we looked at C-peptide levels, so the circulating C-peptide levels in these individuals. And what was interesting is that there was a positive correlation between the plasma EV levels and the C-peptide levels in the individuals with new onset diabetes, shown in red. But this positive association was not in non-diabetic controls. And suggesting that if you have higher C-peptide levels, you are likely to have higher EV-PD-L1 levels in circulation. And so this kind of shows a beneficial role of PD-L1 in preserving beta cell function in these individuals. And then we also looked at earlier time points in the disease course. So we looked at a cohort of individuals with multiple and single islet autoantibody positive individuals and compared with non-diabetic autoantibody negative individuals. And to our surprise, what we saw here was that EV PD-L1 levels were actually higher in the individuals with either single or multiple antibody positive. And then we looked to see if there was a correlation with the number of islet autoantibodies that are present in this individual. And so what we saw that the individuals with single autoantibody positive had a higher trend of EVPDL1 levels in them, suggesting that they might be EVPDL1 might be more beneficial at the earlier time points of type 1 diabetes. And then we also looked at the C-peptide levels, and we saw that there was actually a positive correlation in the EVPDL1 levels with higher C-peptide levels in those individuals. And so coming to our model that we are that from the data we have shown so far, we think that we believe that interferon alpha and gamma induce a PD-L1 level in the cells, and there is more PD-L1 that is trafficked into the EVs. And this EV PD-L1 levels can bind to PD-1 and deactivate or decrease T cell proliferation and activity. And then we showed that with the blocking antibody, we can reverse this effect, wherein when we pre-treat the EVs with the blocking antibody, it led to more activation and expansion of T cells. And finally, we looked at a couple of cohorts of human diabetes samples, and we isolated the EVs from these plasma samples and looked for assayed the PD-L1 levels. We saw that individuals with islet autoantibody positivity had more higher EV PD-L1 levels, and that the EV PD-L1 levels had positively correlated with C-peptide. And this is also published in Diabetology recently. And so finally, there are a couple of limitations to the study. We understand that the EV-PDL1 levels secreted from beta cells may be actually diluted in our plasma samples. So we are looking at the whole EV population, and that the contribution from the islet level may be really, really small. But still, I think it's still reflective about the amount of EVPDL1 levels. The increase we see is reflective about in the individuals. And also, we recognize that the human plasma samples are cross-sectional, and so we need more longitudinal studies to actually see the dynamic changes in the EVPDL1 levels. And so the future directions, we have acquired some periphery samples from human pancreas tissue slices from MPOD. And so we wanted to quantify PDL1 EV levels in these individuals with autoantibody positive and type 1 diabetes and compare them with non-diabetic controls. And also, we are collaborating with wonderful people shown here, Desio Zurich Group, Raghu Mirmura Group at Chicago, and John Pignelli and Pamela Evans Molina's lab to study, or we have developed iPSC cell lines that have actually tagged PDL1 in them to study the physiological impact of beta cell EV transfer to other beta cells and also to other immune cell types. And then finally, we want to understand the changes in the circulating EV-PDL1 levels during T1D progression. And our data suggests that it might be important in the early events of the disease. So we are focusing on early stage of type 1 diabetes. With that, I would like to thank all the wonderful people in this slide, especially my mentor, Emily Sims, who has been a phenomenal and a supportive mentor, and the lab members, and all our wonderful collaborators that I mentioned in the previous slide. And this work wouldn't have been possible without the co-facilities here at IU. and also the study participants and the donors, and the funding. I'm lucky to be funded by Breakthrough 31D and Diabetes Resource Connection. And I would thank all of you for listening to this talk, and thank you.